Assalamu alaikum, dear students. Let's discuss another question which is directly related to the topic bacterial mapping via interrupted conjugation. This is also from the book Genetics, a Conceptual Approach by Benjamin A. Pierce, and its fourth edition is under consideration at the moment. The question states crosses of three different HFR strains with separate samples of an F negative strain are carried out, and the following mapping data are provided from studies of interrupted conjugation. Then the table is given, and the table is showing all three strains HFR1, 2, and 3, and then the genes B, D, C, F, G. They are shown along with the time at which they appear in the F negative cell. Then they asked to construct a genetic map for these genes, indicating their order on the bacterial chromosome and the distances between them. So, two things are asked: order of the genes on bacterial chromosome, and the second thing is distances between them. So, in order to solve, first we have to draw a circle and we will begin with the one strain. It could be HFR1. So for the HFR1, we can see that the B appeared at 3 minutes. So from the time of entry, after 3 minutes, the first gene to appear is B. Then the next gene to appear is D, which appeared at minute 5. So this means that the distance between the two genes is 5 minus 3, 2 minutes. Fine. Then the next gene is C, which appeared at 16 minutes, which is followed by F, which appeared at 27 minutes, and the last one is G, which appeared at 59 minutes. Right now, I'm talking about the genes which are shown or whose data is shown for HFR1. There was another gene E, but it is not shown here for the HFR gene data. So let's start with the B. So the distance between B and D is 2 minutes, and we calculated it by subtracting 3 from 5. Then the next gene to appear is C, and that appeared at 16th minute. So when we subtract 16 and 5, we will get 11, and we will write it in between D and C. Then for the distance between C and F, we will subtract 27 and 16, which will give us 11. We will write them between the two. You can see that they are shown close to each other depending upon the minutes, because there were only 2 minutes difference between the two. So they are shown close to each other. But when we are showing D and C, the distance is further. And the same is true for C and F. Now look at the distance between F and G. This is 32 minutes. So that is why they are written far apart from each other. So we have constructed this circular map based upon the data we received from for HFR1. Now it's time to look for the second strain. The second strain is HFR2. So I have put the first map for reference at the bottom of the slide, and now we will start working with the circular map, and this time the E is the starting point, and from the time of entry till the appearance of E the minutes are 6. And then look at the next gene which is shown, and it is F, and it is a kind of surprise because for our HFR1 E was not there, but there was F. And look at the gene C. The gene C for HFR2 is after F. But when we were constructing the map for HFR1, we were saying that the F is coming after C. So this difference in the arrangement of F and C for HFR1 and HFR2 are indicating that the direction of gene map is different for HFR1 and HFR2. They are anti-parallel to each other. Fine. They are opposite to each other. This word is more appropriate. They are opposite to each other. Okay, Ji. So how we will start? We will start with E, and then the distance between E and F is 14. We will write it there, E and F, and we have placed E in between F and G. Do you remember? फॉर HFR1 हमने 32 मिनट्स का डिस्टेंस बताया था और इन दोनों को काफ़ी फार अपार्ट बताया था. नाउ वी आर प्लेसिंग E इन बिटवीन द टू और हम बता रहे हैं कि E और F के बीच में जो डिस्टेंस है दैट इज 18 मिनट्स. Now look at the distance between F and C. The F and C distance is 11 as calculated, and it is exactly the same for what we calculated for HFR1. ठीक है. फिर उसके बाद C और D के लिए हम कैलकुलेट करेंगे तो वो भी एग्जैक्टली 11 है जैसे कि हमने HFR1 के लिए कैलकुलेट किया था. तो इसका मतलब है ये पोजीशनिंग हमारी बिल्कुल ठीक है. F to C, F to D, and then when we will calculate between the D and B, we will get 2, which is exactly similar to the HFR1 data. So F, C, D, and B, whether we look at them from this direction जैसे कि HFR1 में था B to D, D to C and C to F, the distance between the gene is 2, 11, 11. And when we are doing it in the opposite direction from F to C, C to D and C to B, they are again 11, 11, 2, which are similar to the data calculated for the 1. So there is similarity for these two. Now it's time to look at third one. And the third one is HFR3. The genes are given in the direction D, C, F, E, G. Fine? D, C, F, E, G. So this sequence is similar to the sequence for HFR1. Fine? और इसको अगर हम calculate करेंगे the distance between D and C. So this is the first gene to appear and the second gene is C. Upon calculating the difference, 11 is identified. So yes, this is the one which was verified for HFR1 and 2 as well. Then for the second one, C and D, C and F, the difference is 11 minutes. Again, verified. But when it comes to E and G, let's see F and E first. F and E difference is 18 minutes. which was verified for HFR2. ठीक है, HFR2 में भी E और F के बीच में 18 था और यहाँ पे जब हम F से E के बीच में देख रहे हैं तब भी 18 मिनट आ रहा है. तो इसका मतलब है इनकी पोजीशंस भी वेरीफाइड होगी. अब जो लास्ट की कहानी रहेगी बिटवीन E एंड G, तो E एंड G को जब हम देखेंगे तो इनके बीच का जो डिफरेंस बन रहा है दैट इज 14 मिनट्स. तो ये मिनट्स हम लोगों को हमने वैलिडेट कर लिए हैं और वो वैलिडेशन हमें बता रही है कि yes, the genes are present in this particular manner between on that bacterial chromosome. So इस तरह हम लोगों ने इसको सॉल्व कर लिया बैक्टीरियल मैप को और अब हमें पता चल गया है कि हमारे पास जो ट्रांसफर्स हैं द ट्रांसफर्स में से हमारे पास दो ऐसी हैं जो कि एक डायरेक्शन में ट्रांसफर हो रही हैं जबकि जो थर्ड वन है वो अलग डायरेक्शन में ट्रांसफर हो रही है. सो ये वो थोड़ा सा इसमें टाइपो एरर्स हैं तो आप इसको करेक्ट कर लीजिएगा. हमारे पास HFR1 और HFR3 सेम डायरेक्शन में मूव कर रही हैं जबकि HFR जो 2 है वो हमारे पास ऑपोजिट डायरेक्शन में मूव कर रही है. 1 and 3 इन द सेम डायरेक्शन एंड 2 इन द ऑपोजिट डायरेक्शन. थैंक्स फॉर लिसनिंग. अगर कोई भी क्वेरी है इससे रिलेटेड काइंडली मुझे ज़रूर बताएं ताकि हम इसको दोबारा से डिस्कस कर लें या फिर किसी और एंगल से डिस्कस करें ताकि क्लैरिफिकेशन आसान हो जाए. ताफिज़.